Funding for Yale Cancer Answers is provided by Smilo Cancer Hospital. Welcome to Yale Cancer Answers with Dr. Anees Chagpar. Yale Cancer Answers features the latest information on cancer care by welcoming oncologists and specialists who are on the forefront of the battle to fight cancer. This week, it's a conversation about hematopathology and breast cancer research with Dr. Samuel Katz. Dr. Katz is an associate professor of pathology at the Yale School of Medicine, where Dr. Chagpar is a professor of surgical oncology.
So, Dr. Katz, maybe we can start off by you telling us a little bit more about yourself and what it is you do.
Sure. So I'm a physician scientist within the Department of Pathology. I split my time where I spend 20% on clinical service, diagnosing blood cancers, leukemias and lymphomas. But I spend the majority of my time running a basic research laboratory that focuses on questions of how cells die. And we approach it from two different standpoints. One, by the pathway within the cells that cause them to die, but then also by a pathway external to the cells and how we can kill them. Because if we can manipulate the ability to kill cells, that could help in many different diseases like cancers.
And so, tell us a little bit more about how you kind of came to work on breast cancer as a hematopathologist. You mentioned that in your clinical role, you really focus on blood cancers. So how do you get into the breast cancer world?
Yeah, so as a hematopathologist who focuses on the blood and the blood system, I got very interested in a particular cell type called a T cell. And T cells are important in our immune system to attack cells that have been infected with foreign agents. They're able to recognize the cells as being infected and kill them. And people have realized that they have such incredible ability to kill those infected cells that perhaps we can usurp that ability in order to attack other cells like cancer cells.
And so tell us more about how this kind of works in breast cancer and more about your research.
Sure. So in breast cancer, as well as many other types of cancers, there are proteins that are on the surface of the cell that are not present in normal cells. And so we have devised a protein that we can add into the T cells called a CAR, or chimeric antigen receptor, thus making a CAR T cell that can recognize this aberrant protein on the breast cancer cell and direct the T cell's killing ability towards that breast cancer cell.
And so that sounds really fascinating. So tell us more about how CAR T therapy works. I know some of our listeners may be familiar with this, but many may not be. So, how do you actually change these T cells to make them recognize these proteins on the surface of the cell? Because it sounds like essentially what you're doing is you're taking a patient's immune system, these T cells, and you're kind of giving them a GPS, a targeting system to say, go after those cells, those cancer cells. But somehow you have to get the GPS into those T cells. How do you do that exactly?
Absolutely. And so there's a number of ways in which to as we say, reprogram those T cells. The most commonly used ones are viral approaches using retroviruses or lentiviruses, where a piece of DNA in that virus will infect the cell and then integrate or become part of that cell's genome or DNA. And it'll then express this new protein that we've made, which we can discuss later, called the CAR, or the chimeric antigen receptor. Another way in which to do it, which is the approach we've taken and really came about because of some work by a senior professor here at Yale called Sherman Weissman. He kind of took me under his wing as a mentor in this approach where instead of using DNA, he was using RNA. And so we can take the T cells out of the patient and what we call electroporate in order to give them kind of a little shock that gets the RNA into the cells. And this has a very high efficiency of being able to reprogram those cells using the RNA in this manner. But it also has a lot of other advantages, chief among them being safety, in that when you put an RNA into a cell, it doesn't change the genome at all of the T cells that you're taking from the patients. It only makes that RNA, which then makes that protein, and after a period of time, it goes away. And so there's an added safety to that.
And that also sounds like that would be particularly handy once the job of getting rid of this cancer is done, that the cells go back to normal. So how long does it take for that RNA to disintegrate or go away?
Right. So the RNA actually is very short-lived, but the protein it makes can last a little longer. And it really depends on the particular protein that you're making, but we see it in the order of about a week or so. So one could envision giving this therapy as a weekly type of basis where you're giving these cells that have been reprogrammed with RNA, or in newer work that's still ongoing, trying to actually deliver the RNA into the body without having to take out the T cells to reprogram them.
And so, it sounds like it really is intriguing, right? That you kind of give these T cells a little shock, give them an RNA to make a protein. That CAR protein, goes and attacks these cancer cells in a very specific way, because presumably this protein is found on cancer cells and not on normal cells. So where are we in terms of actually getting this into clinical trials?
Yeah, so we're still in the early phases, I'd say, of doing this. There's a lot of work to be done to optimize the system overall. And these include things that improve the ability of the T cells to kill, to make sure that they don't get exhausted, to make sure that, again, as we're saying, to really make sure that's safe. We still have work to do in animal models before we can get it into the clinical sphere. But because of the RNA approach and the safety, we do think it is a easier transition to getting it into patients.
And in terms of, the safety and side effects, can you talk a little bit more about the side effects? I mean, I would assume that this has a lot to do with whether these proteins are on normal cells in any capacity, or whether they are really 100% only on cancer cells. And also revving up the immune system, you might think that you might get some immune-related side effects as these T cells go about doing their business.
Sure. And so maybe best to do is to take one step back and to say where the CAR T cells have been really successful to date in the clinic. And these have been against actually targets that are on B-cell malignancies or leukemias and lymphomas. And they're going after a target called CD19, which is expressed on the surface of those B cells. And that really is unique to those cancer cells, as well as normal B cells. And so when the CAR T cells are introduced to those patients, it does get rid of all their normal B cells, but patients are fine with that. We can live without our B cells. There are some side effects that is seen with that therapy. One is called a cytokine release syndrome, where because you're getting so much killing so quickly of the cancer, it releases a lot of these cytokines that leads to kind of like an immune storm within the patients. They feel very sick, and you have to really watch them carefully within the hospital. And there's also been some less well understood neurological disorders that occur in some patients. And some people have hypothesized that might be due to the fact that we learned later that there's a cell type within the brain that has very low expression of this target. And so then that gets us back to breast cancer and solid tumors, where there aren't as many great targets that we know of that are uniquely expressed on the surface of these cells. The one that we're going after actually turns out to be increased in more than half of triple-negative breast cancers, and its expression correlates with poor prognosis within these patients. There is some very low expression during development, but we have some reasons to believe that we can kind of thread the needle between this very high expression on the cancer and this perhaps low expression on some normal tissues.
Yeah, I mean, I think that in general, for most cancer related drugs, there is this, it's never completely black and white. Even chemotherapy, we know we still use, and it really is designed to attack rapidly growing cells and dividing cells, but you still get some normal cells that are also rapidly dividing, like your hair, for example, which is why many patients undergoing chemotherapy lose their hair. So it sounds like even if there was a potential differential there, it still might be really handy in terms of a therapy, especially if it was less toxic than our standard therapies, which for triple-negative breast cancer are primarily chemotherapy. Now, the other question that I have for you is, in triple-negative breast cancer in particular, we've seen that there are now therapies that are being used that are immunotherapies. So really therapies that are designed to unleash the immune system, especially because some of these triple-negative breast cancers, they tend to evade the immune system. So if that's the case, and this CAR T therapy is really designed to use the immune system, is it the idea that this would be paired with immunotherapies, or are you thinking about a different way of attacking this?
So I think there is a potential for testing the two together, but it is very different in the way these two different classes of immunotherapies work. So the ones that you're referring to, so-called checkpoint inhibitors, these are ones that rely on new antigens that are made within the cancer cell that are mutant and specific to the cancer cells, and they really are unique. The T cells use their normal T-cell receptors to recognize those, but there's a so-called brake mechanism that prevents the T-cell from killing, and the checkpoint inhibitors take away that brake. The CAR that I've been talking about, these CAR T-cells, this is a new protein that we've devised by taking pieces of various other parts of the T-cell receptor and other antigen recognition domains. And they recognize, or we've designed this one to recognize a specific protein that's not mutated, but wild type. And this then activates the CAR T cell rather than stopping the brake. I'd say it's more akin to pressing on the gas pedal when we have that specific protein.
Got it. Well, we need to take a short break for a medical minute, but please stay tuned to learn more about the role of pathology and new research into a potential target for metastatic triple-negative breast cancer with my guest, Dr. Sam Katz.
Support for Yale Cancer Answers comes from Smilow Cancer Hospital, where their prostate and urologic cancers program provides a multi-specialty team dedicated to managing the diagnosis, evaluation, and treatment of bladder cancer. SmiloCancerHospital.org. The American Cancer Society estimates that more than 65,000 Americans will be diagnosed with head and neck cancer this year, making up about 4% of all cancers diagnosed. When detected early, however, head and neck cancers are easily treated and highly curable. Clinical trials are currently underway at federally designated comprehensive cancer centers, such as Yale Cancer Center and at Smilow Cancer Hospital, to test innovative new treatments for head and neck cancers. Yale Cancer Center was recently awarded grants from the National Institutes of Health to fund the Yale Head and Neck Cancer Specialized Program of Research Excellence, or SPORE, to address critical barriers to treatment of head and neck squamous cell carcinoma due to resistance to immune, DNA-damaging, and targeted therapy. More information is available at yalecancercenter.org. You're listening to Connecticut Public Radio.
Welcome back to Yale Cancer Answers. This is Dr. Anise Chagpar, and I'm joined tonight by my guest, Dr. Samuel Katz. We're talking about the role of pathology and some new research into CAR T cells, but now for a new indication, and that's really metastatic triple-negative breast cancer. So, Dr. Katz, I want to go back to something you were mentioning right before the break, which is how traditional immunotherapies, these checkpoint inhibitors, which we now use in triple-negative breast cancer, really kind of get rid of a break, as you phrased it, in terms of T-cell killing, right? Because we know that certain cancer cells, especially triple-negative cancer cells, may kind of put a break on those T cells to kill off these cancer cells. And so traditional immunotherapies will remove that break. Your CAR T therapy is more like an accelerator, finding a new target on these T cells to kind of attack cancer cells in a different way. So kind of like putting on an accelerator. My question is, how do those two work together? Or is there an interplay? Thinking about, driving a car, if you step on the gas while you're still got a brake on, it generally doesn't work very well. Can you talk a little bit more about that?
Absolutely. And I think that's why, as you kind of suggested, the combination of this might be very useful because while if you're just releasing your foot off the brake by using these checkpoint inhibitors, if you don't have something driving, if there isn't a mutant antigen for you to go after, then the car won't move forward. The T cell won't kill. On the other hand, like you said, if the CAR T cell is engineered so that it's always pressing on the gas pedal, yeah, it might try going forward. But if you have that brake present at the same time, then it won't be able to. But if you can manipulate the cell in ways that many people are to kind of combine the two, then perhaps we could get the full benefit of this. I also want to bring up one other thing that you had mentioned before the break, which is kind of getting towards the difference between solid tumors like triple-negative breast cancer and the blood tumors where CAR Ts have worked so well. Solid tumors have remained a real challenge for the CAR T field to be able to work efficiently. And that's because they create this tumor microenvironment that kind of quells the T cell, some of which might be to increase the brake like we've been talking about. Another way is you can imagine that the car won't do so well if you're always pressing the gas pedal, right? You'll run out of gas eventually. And a lot of the CAR T designs in the past have this problem where you're always pushing on the gas, even when when you don't want it to, when you don't have that target in sight. Fortunately, some work in the lab by Po-Han Chen, another physician scientist who's been working on this problem, came up with a new design towards our car to make it so that it only presses on the gas when we want it to.
That's interesting. Can you tell us a bit more about that? I mean, one would think that if there wasn't a target, that the T cells really wouldn't have anything to go after. And so they would just be kind of floating around looking for that target if it should appear. So how do you turn on and turn off these T cells so that they don't get overly active and exhausted, as you put it?
Yeah, that's a great question. And I think what we have to remember is when we're putting in this CAR, this chimeric antigen receptor, it's really a man-made Frankenstein type molecule. It hasn't been engineered by nature over, millions of years of evolution. It's something that we've come up with and made in the lab. And so therefore it doesn't work necessarily perfectly. We've taken snippets of different proteins and put them together. And a normal receptor that's on a cell will only single to have its downstream effects when it engages its target. But these CARs that we've made ourselves, they have a little leakiness to them, many of them. And that leads to what we call tonic singling all the time, or pressing on that gas pedal all the time. And Pohan has realized that one of those domains could be optimized to help reduce that issue. And I think that's going to be really critical for when we start targeting solid tumors.
And so when you say optimized, do you mean like it's kind of got a way that it learns when to turn on and when to turn off? Because presumably you want the thing to turn on when there is that target, and you want it to go full speed ahead and kill that target. And when the target isn't there, well, then you want it to conserve its energy and lay low for a bit.
Right. So looking at the actual structure, or the presumed structure of the molecule, we hypothesized that they might be coming together. So the singling usually occurs when you get more than one of these CARs coming together, being brought together. And that's what happens when it engages its target on the other cells. And so by changing one of those domains that we thought was leading to that aggregation and that baseline single, we were able to decrease that baseline singling and make it so that it only singles when it really is being brought together by the antigen on the other cell, and not when it's existing on its own in the T cell.
Got it. The other question that I have for you is, you mentioned that one of the things that makes solid tumors tricky is this tumor microenvironment. The fact that, the cancers know how to make an environment around themselves that's very comfortable for the cancer cells to grow in, and not so comfortable for anything else to kill them. But in thinking about CAR T therapy and blood cancers, when you think about metastatic disease, really, there is potentially a way to think about solid tumors that may be like a blood tumor, in the sense that when they're metastatic, you're really trying to get at the circulating tumor cells and the disease that isn't necessarily in a particular solid organ. Can you talk a little bit about that? Is CAR T therapy particularly good for metastatic disease and reducing the circulating tumor burden?
Yeah, absolutely. So as I was mentioning, the CD19 CAR that targets B-cell leukemias, that one works phenomenal. It doesn't have any of that, by the way, the tonic singling that we were just talking about. It is a great target. It's all in the bloodstream, and patients do very well with that. Just underneath that, there are so-called B-cell lymphomas, which take up residence. They form more of a mass as opposed to just being circulating through the bloodstream. That they also can use the CD19 CAR, and they do okay. Not as well as the leukemias with that CD19 CAR, but still somewhat okay. And part of that is probably this tumor microenvironment that's created there. Now, one of the best reasons to use the T cell or to deliver these CAR T cells, is that the T cells seek out and destroy these metastases that are throughout the body. There are molecules that kind of tell them to look within these areas, and it gets them places where other less smart drugs might not realize how to get to or where to go to. And so improving CAR T cells' ability to find these metastases is another active area of investigation. In fact, we have a collaboration with another senior professor, John Morrow, in determining ways of how we can improve the T cells' ability to traffic, to get to where they're going to. And then once they're there, they have to then face this kind of a barrier, this impenetrable barrier that the tumor kind of forms this wall. And so there are other ways that people are designing to equip the T cells to kind of get through that barrier a little bit more as well.
As you mentioned, thinking about metastatic sites and so on, and the ability for T cells potentially to navigate through these barriers better than other drugs, it makes you think about things that have been historically very difficult for us to treat with standard chemotherapy. And that's kind of getting to brain metastases and getting past the blood-brain barrier. But earlier, before the break, you were talking about some neurotoxicity associated with these newer therapies. Can you talk a little bit about whether CAR T therapy, you envisage this really having a role to play in brain metastases and how exactly that would work?
Yeah, absolutely. So interestingly enough, some of those original patients that had leukemias or blood lymphomas wound up having disease within their brain. And it was found that the CAR T cells were making their way in there and actually fighting off the disease that was there. So I think the potential is possible. And it's not quite understood yet whether they were able to get in because the blood-brain barrier that we talk about was disrupted a little bit because the disease was already there, or whether the CAR T cells are able to, even in a completely intact blood-brain barrier, get in. But I think there certainly is the potential. And there have been several studies since then trying to target not just hematopoietic tumors that make it to the brain, but also solid tumors that have made it to the brain as well, in addition to brain tumors themselves, where there are different CARs that people have been developing in order to do that. And there is some evidence of some efficacy. Still needs to be improved, though.
Yeah. It sounds like such a wonderful, exciting new target, but I wonder about the downsides as well. So, when we think about really turning on the immune system, after having lived through the COVID pandemic, many of us saw that there were some patients whose immune systems were turned on so much that you ended up with this immune storm. And really that caused a lot of side effects for these patients. Would you expect the same kind of thing with CAR T therapy? I mean, it seems like it might be a balance between too much and too little. On the one hand, you don't want your T cells to get exhausted. On the other hand, you don't want them overly working too hard either at the expense of toxicity.
Absolutely. And this is one of the reasons why I really appreciate the wisdom of Sherman Weissman in devising and thinking about this RNA approach. So when you give a standard CAR therapy using the lentiviral type approach in DNA, you really don't have any control over those T cells and how much they proliferate, how long they stay around for, what kind of dosing you give. And if a patient winds up having some of these side effects, there's not much you can do. On the other hand, for the RNA approach, you can very precisely decide how much you're giving and when. And you can titrate that amount so that you can make it less in order to not get into that territory where you get those types of side effects.
Dr. Samuel Katz is an associate professor of pathology at the Yale School of Medicine. If you have questions, the address is canceranswers@yale.edu, and past editions of the program are available in audio and written form at yalecancercenter.org. We hope you'll join us next time to learn more about the fight against cancer. Funding for Yale Cancer Answers is provided by Smilow Cancer Hospital.